Now we're going to move into Chapter 9, Part 2, where we discuss muscle mechanics. There's several points or bullets that I want to make here when we talk about whole muscle contraction. So number one is that the same principle applies to the contraction of a single fiber or muscle fiber cell and the whole muscle. So here we have Z disc to Z disc, which is sarcomere. When the sarcomere contracts, and you have several of them contracting, right? When the sarcomere contracts, the myofibril contracts. When the myofibril contracts, the muscle fiber cell contracts. When the muscle fiber cells contract, remember back to fascicle, the fascicles contract. When the fascicles contract, the whole muscle contracts. The second bullet point here is that contraction produces muscle tension, and this tension is what we use to move the load or move the object. The third point that we need to talk about, it says contraction may or may not shorten the muscle. Now I know up until now we've been saying contraction is shortening of the muscle, but there are some instances where the muscle does not shorten. And here is that example. So isometric contraction, this means same, metric means length, right? There's no shortening of the muscle. Muscle tension increases but does not exceed the load. So say you're trying to lift a car. You have your hands on the bumper, you're pulling with all your might, you're generating tension in your muscles, but you cannot generate enough muscle tension in order to exceed the load. So in this case, the muscles are not shortening. Isotonic contraction, the muscle shortens because the muscle tension exceeds the load. This is what we would think of with normal contraction. There are two subtypes. We can look at concentric contraction. The muscle shortens and it does work. The best example of this would be flexing, right, or flexation. Eccentric contraction, the muscle contracts as it lengthens. A good example for this would be extension. So think of concentric as you're lifting the weight up. The biceps brachii is shortening and it's doing work. That's concentric contraction. If you have the same weight in your hand and you're lowering the weight back down, now the muscle is contracting, it's producing tension, but it's lengthening as you go to put the barbell down. So think of concentric as when you flex, and eccentric as when you extend. And if you do this, physically do this, go pick up a weight of some sort and do the concentric flexing and the eccentric, you'll see in the eccentric that your muscle is still bulging. It's still contracting, but it's contracting as you lengthen. Fourth item here, we need to talk about the force and duration. So this duration means how long, right? Force, think of strength maybe. Contractions vary in response to stimulus. So you can have a change in stimulus frequency. So how many times, right? How many times is the muscle stimulated? Strength, how forceful is the stimulus? Before we look at some examples of stimulus frequency, we have to go back and talk about something called a muscle twitch. A muscle twitch you've already seen before. You've seen the action potential generated as it goes from resting membrane potential and then eventually back to resting membrane potential. So a muscle twitch is the simplest contraction resulting from a muscle fiber's response to a single action potential. Simplest contraction that's observable. Here's a myogram. They're showing you a twitch. So this is what a twitch looks like. Here's a single stimulus. So this is the neuron. We have depolarization, we have an action potential, and we have repolarization. So we've already seen this, right? Here's the period of contraction where you think of action potential. Here's the period of relaxation. We're trying to go back to our -90 resting membrane potential, right? Another example of a single twitch. So here's the stimulus. We have contraction, we have action potential, we have relaxation. Okay, so let's look at a change in stimulus frequency. So what happens if we have an increase in stimuli? You can see here at the bottom there are 4 stimuli. This would be a neuron telling a muscle to do something, right? So another stimulus is applied before the muscle relaxes completely, and this results in building tension. This is how our muscles normally work. So this is normal behavior for muscles. They call it unfused or incomplete tetanus. Tetanus term has to do with tension. This is not the same as the bacterial infection that causes lockjaw. So we can see the first stimulus is sent. We start contraction, we start to relax, and then another stimulus is sent. We start contraction, relax, another stimulus is sent. And you can see that complete relaxation doesn't occur. The whole time that we're sending these stimuli, we're building tension in the muscle. So let's look at another example of stimulus frequency. This is called fused or complete tetany or tetanus. And you can see tension is building just like in the other model, but you can see that there's no relaxation. And there are many more stimuli, so an increased number of stimuli over a shorter period of time. So higher stimulus frequency, no relaxation between. So we can see the muscles start to contract, and then continual stimuli are applied and there's no relaxation. And we stay at this plateau or fused complete tetanus. This doesn't happen very frequently. This would lead to eventual muscle fatigue. So what will happen is you can only do this fused tetanus for a short period of time. I think your book mentions like some of those superhuman feats where there's a car on top of a child, somebody comes over and lifts the car. You can exert this much tension for an extended period of time, but only in extreme circumstances, and then eventually you're going to have muscle fatigue and you're going to drop to zero tension. Now we're going to look at stimulus strength. So twitches can vary in strength. We're not looking at the number of stimuli, but we're looking at the tension that builds and how long it stays in a particular muscle. So differences in strength and duration of twitches are due to the different metabolic, so breakdown and build up enzymatic properties in muscles. So here's an eye muscle. This is your calf, and this is your soleus, which is directly underneath your calf. So we can see a single stimulus, and we're looking at over time, and we're looking at the amount of tension. So you can see that the eye muscle works very quickly. You can see that the gastrocnemius muscle stays contracted over a longer length of time. And then you can see that the soleus, this stays contracted and develops a larger amount of tension over a longer period of time. Stimulus strength is dependent on motor units. So the motor neuron and all of the muscle fibers that innervate are called a motor unit. So you can have small motor units that have 20 or so muscle fibers. You can have large ones that have thousands. So the stimulation of a single motor unit will cause only a weak contraction of the entire muscle. This figure, we can look at 2 different motor units and see that motor unit 1 comes and forms 2 neuromuscular junctions, and that motor unit 2 comes out and creates 3 neuromuscular junctions. So a motor unit is the motor neuron and all of the muscle fiber cells that are innervated by that nerve. You can see the variability here. Our brain could send a message out and excite 2 muscle fiber cells. Our brain could excite 3 muscle fiber cells, or our brain can send a signal on both motor unit 1 and 2, and we could have 5. So this would determine the strength of a contraction, depending on how many motor units are being activated and how many muscle fiber cells are responding. We use this term when we talk about recruitment. So basically, motor units being recruited, how many are being recruited? Motor units in a muscle usually contract asynchronously. Basically, they don't contract all at the same time. Some contract and others relax so that you can have continual contraction. Helps prevent fatigue. This figure is trying to show you how stimulus strength corresponds to the number of motor units. What they're saying is if only one motor unit was activated, it might not be enough to reach the threshold stimulus. If all of the motor units are activated, that's enough to have maximum contraction. Last topic is to talk about muscle tone. So with muscle tone, your muscles are staying in a slightly contracted state. So if you have flabby muscles, they're not slightly contracted. If you have muscle tone, they're in a slightly contracted state. They're ready to do work. They're firm, healthy, ready to respond muscles. And we're going to talk about some of these reflexes later, but I wanted to show you that there are nerves that come into the muscles and they tell your brain information about the position of a muscle, how stretched a muscle is, and also to the tendons down here. So your brain is constantly sending and receiving messages from your muscles. Remember that we need lots of ATP for muscle contraction. So in order to cause the myosin head to extend into the cocked position, for it to detach, to pump the calcium back, to operate the sodium-potassium pump, we store a little bit of ATP in our cells, but that is depleted within the first 4 to 6 seconds. So we need other ways to generate ATP. These are the 3 ways that we can generate ATP. Something called direct phosphorylation, anaerobic, this means not with oxygen, and then aerobic, which means with oxygen. With direct phosphorylation, we're using something called creatine phosphate. It has a phosphate group on it. And we're taking an enzyme called creatine kinase. And how do I know it's an enzyme? It ends in ASE. So we take the creatine phosphate, the enzyme creatine kinase pulls the P off, and if I add ADP plus P plus energy, I'm creating ATP. So this is about the 6 to 15 second mark that this creatine phosphate is going to power the cell. In the anaerobic pathway, this does not require oxygen and only 2 ATPs are generated. So you're going to go to the first step, which is called glycolysis. So we are taking glucose, we're starting to break it down, but we only get out 2 ATPs if there's no oxygen. In the aerobic pathway, we're going to produce lots of ATP, 36. It requires oxygen and it occurs in the mitochondria. Here's a figure from a general biology book, but I like the simplicity of it. So here is the aerobic pathway. There are 4 steps to the aerobic pathway. We're not going to look at those in detail, but aerobic pathway, you start with glucose and you end up with 36 ATPs. You have to have oxygen and you have to have mitochondria. Anaerobic pathway, you only make 2 ATPs and you only do the first phase of the aerobic pathway. This does not require oxygen and you make 2 ATPs total. So what's the big picture? The first 4 to 6 seconds, you use your stored ATP. Then the 6 to 10 second mark is when you're stealing phosphate from creatine phosphate to make ATP. Then we're going to use our stored glycogen. We store glucose in our muscles, and we're going to use that and turn that into ATP. As we go through this exercise, we are switching between aerobic and anaerobic back and forth. So aerobic, you're breathing fine, you're getting tons of oxygen, you're making tons of ATP. Then we might switch into anaerobic. If you get to the point that you're really sucking wind, as I call it, right? You're that is anaerobic. You're probably only producing a little bit of ATP to kind of get you to the next moment, Maybe you start running a little bit more slowly, you catch your breath, and then you can go back into aerobic. Muscle fatigue is the inability to contract, and you can see here that a total lack of ATP rarely occurs. Remember, ATP is necessary for neuron transmission as well as muscle, and so it would be bad if we were completely out of ATP. But what usually happens is we have some sort of ionic imbalance. So something happens to your potassium level, your calcium level, your phosphate level, and it interferes with the electrical depolarization and repolarization of your muscle fiber cells. We used to call this oxygen deficit. The book has a new term, excess post-exercise oxygen consumption. The basic question here is, why are we breathing deeply after we work out? Well, we've stopped making ATP, right? We don't need it. We're not running anymore. Why are you still breathing heavily? Well, because you have to replace all the reserves that you stole in the beginning. So we stored oxygen in our muscles. We have to put that back. We have to replace the glycogen. Remember, this was the glucose that we stored. We also have to replenish the creatine phosphate. We have to add phosphates back to the creatine so that it'll be there next time we need it. We also need to put some ATP back in our muscles for the next time. So there's lots of reasons that afterwards you're breathing deeply, you have to replace all of the stores or reserves that you stole from to get you to the end of exercise. 40% of the energy released in muscles actually does work. The remaining 60% is given off as heat, and this helps us maintain our body temperature. We learned about this in Chapter 1 when we talked about shivering and sweating. So what are the factors of muscle contraction force? So the forcefulness, or the amount of tension created, can be dependent on a number of things. So number one here, I have the number of muscle fibers stimulated. So recruitment. We said the more motor units are recruited, the greater the force or the tension is developed in the muscle. We can also look at the size of the fibers. If you have really large muscle fibers, they can develop more tension, therefore more force. Number 3 says frequency of stimulation. How many stimuli? Remember, we talked about the complete and incomplete tetany. So depending on the number of stimuli depends on the tension that's created in the muscle and for how long. The fourth is the first one that we haven't really discussed yet, and that's the degree of the stretch of the muscle. So how stretched were the muscles before contraction? This figure is showing you the optimal sarcomere length. So if the sarcomere is at rest and it is at this length, it can generate the most tension. They're saying if the sarcomere is stretched too far, the myosin head can't really reach up and pull the fin over. So excessively stretched is bad, and greatly shortened is bad. So in this case, can you see how the myosin heads don't have much to grab? They can't pull the thin myofilament over because it's already completely overlapped. So there is an optimal length that we want the sarcomere to be in order to generate the most tension. Velocity and duration of contraction. So velocity is speed and duration is length of time. So how fast a muscle contracts and how long it can stay contracted is influenced by these 3 factors. We've already seen recruitment and we've already talked about load, so we're going to learn something new about muscle fiber type. There are different muscle fiber types, and they're categorized by 2 different characteristics. I'm going to let you read over this, and I'm going to go to the figure to explain it. Here are the 3 types. There is a slow oxidative fiber. These are usually red in color. They have tons of mitochondria. This should tell you something about ATP production. They have tons of stored oxygen, and they have capillaries that give them the red appearance. These are great for aerobic respiration, so that's producing those 36 ATPs, and they are fatigue resistant. The example would be like the soleus, which is behind the gastrocnemius. The soleus is the muscle that you would use if you're running a marathon. Your back muscles, remember that you're standing erect all day long, and the muscles that make up your back or posterior back muscles need to be contracted for long periods of time. So the muscle fibers themselves, the genetics of the muscle fiber cells, tell them to be slow oxidative. Fast glycolytic, these are white in appearance. They have few mitochondria, so that means less ATP, right? They have fewer myoglobin, so that means less oxygen. These are adapted for quick responses. They are not fatigue resistant. It's where we just need a burst of energy. So the gastrocnemius would be the muscle you're using if you're sprinting. Your eye muscles, orbicularis oculi, it just needs to open and close, right? And then there's an intermediate, and that is fast oxidative. They vary from red to pink, and they're adapted for medium intensity activities. So sprinting, walking. So this table is great at looking at the differences. Let's look at the two extremes. Here's slow oxidative. It uses aerobic, and it is fatigue resistant. Fast glycolytic, it uses anaerobic pathway, and it is fatigable or fast to fatigue. So these would be muscles that would need like long endurance muscles if you're running a marathon, and these would be the muscle fiber cells that you would have a quick response. So these are genetic, right? Somebody could be a born sprinter. That might mean that they have more fast glycolytic muscle fiber cells in their gastrocnemius. Now we move on to load. We're still talking about speed and time, right? Velocity and duration. This is kind of a no-brainer. If you have a heavy load, you can't hold it for very long. Your muscle cannot contract and hold that heavy load for as long as it could hold a light load. We've already looked at the influence of recruitment, and we can see that as more motor units are excited, we have maximal contraction. So what are the effects of exercise? What happens if we're doing lots of aerobic exercise? Well, the muscle fiber cells can change or adapt. So the muscle fiber cells will increase capillaries, increase mitochondria. Why? Because you need mitochondria to make ATP. They're going to store more myoglobin. They can store more glucose. So basically, you're changing the environment of the muscle fiber cell when you work it out aerobically. And there's also an interesting concept here that some of these fast glycolytic fibers can adapt or change into those intermediate oxidative fibers. So we can kind of convert some of our fibers, even though they're based on genetics, the environment does have an impact on them. So what about resistance exercise? So like lifting weights. Basically what's going to happen is the myofibrils are going to grow in size, and then eventually they're going to split. So it's not that the entire muscle is growing, it's that the myofibrils inside the muscle fiber cell are growing, and it's making the muscle size seem larger. And then obviously if we have more myofibrils, we have more sarcomeres, we have greater tension developed during contraction. We need to briefly discuss the overload principle. So forcing a muscle to work hard promotes increased muscle strength and endurance. So this is that no pain, no gain, right? You have to work the muscle to the point of fatigue in order for it to change and develop some of these characteristics that are beneficial. We should focus on the word atrophy here. So muscles must remain active to be healthy. Atrophy is when you have degeneration of a muscle. It could be due to the fact that you're not using it, or it could be due to the fact that the neural stimulus has been cut, or there's been some sort of neural loss to that particular muscle. So muscle, if you don't use it, you lose it. 5% decline per day. Paralyzed muscles are going to atrophy. So basically you need to stay active in order for your muscles to be healthy. There are some wonderful tables to look at in the book that break down the differences between skeletal, cardiac, and smooth. I'm going to move straight into the slide where I talk about cardiac muscle, but please look at these tables. We've been talking about skeletal muscle. Now I want to switch gears and talk a little bit about cardiac muscle. So remember, cardiac muscle is involuntary. The cells are shorter and thicker and they're branched. They have electrical gap junctions. They're going to have some differences, especially in terms of calcium. So the majority of the calcium needed for contraction comes from extracellular, so outside when we talk about cardiac muscle. They repair by fibrosis, so they do not completely regenerate. So remember, fibrosis was scar tissue. And then they use aerobic respiration. That is oxygen needed yielding 36 ATPs. They are very vulnerable to interruptions in oxygen supply because they use aerobic respiration almost exclusively. Now, smooth muscle is found in the walls of most hollow organs. It allows for something called peristalsis. This is alternating contractions in order to move something through a lumen, which is an opening. So think about a blood vessel. The space inside, that's the lumen. And if I wanted to move fluid from one area to the next area, you can't just do contraction. The food would stay right here in this area. So you're actually doing peristalsis, which is alternating contractions and relaxations that squeeze a substance through a tube. We'll see some examples, and people like to think of it as the wringing of a towel. That seems to be the best example to understand or illustrate smooth muscle action. There are no neuromuscular junctions. Instead, these are going to be controlled by autonomic nerve fibers. And this is also involuntary. They can contract without nerve stimulation. So by stretch, so say the bladder, your bladder filling, once it reaches a certain size, the muscle is going to contract and allow for the expulsion of urine. Smooth muscle is named because it looks smooth. So it forms these spindle-shaped fibers. There's one nucleus, there's no visible striations or lines, there's no sarcomeres, there's no Z disc. So basically there's very little sarcoplasmic reticulum, no T-tubules, and the calcium comes from extracellular fluid. Here is the relaxed smooth muscle fiber. Here is the contracted smooth muscle. You can see that these dense bodies attached to the myofilaments cause this squeezing or wringing motion. Smooth muscle contracts slowly and contracts altogether. They're prolonged contractions. They're very energy efficient. They use much less ATP than we need for cardiac or skeletal. One of the special features of smooth muscle is this kind of stress or stretch relaxation response. So basically, when smooth muscle is stretched, it's going to briefly contract and then it's going to relax. This allows organs to have a huge volume change without becoming flabby when they're relaxed. So basically, it adapts to large volume versus low volume without the muscle becoming flabby so that it won't work the next time. So it basically doesn't allow for this area to be too distended or to stretch out of shape. It adapts to the new shape. And you could see that this would be important if we're going to store something in the bladder. So imagine if your bladder filled up and then you released the urine from your bladder, but it stayed the same size empty as it did when it was full. That wouldn't be a great storage organ for us, right? It also has the ability to contract on demand. So as soon as food gets pushed from our palate into our esophagus, it brings on the peristalsis or alternating contractions of our esophagus to push food down to our stomach. Last special feature is that smooth muscle can be regenerated. It does not repair by fibrosis like cardiac muscle does. So not only can smooth muscle divide, which is mitosis, it can increase in size or in number, that's hyperplasia. The next few slides are going to talk about developmental aspects. I just want to point out the ones that are important. Cardiac and skeletal muscle become amitotic. This means no division, but can lengthen and thicken. Smooth muscle regenerates throughout your lifetime. Female skeletal muscle mass is about 36%, where males is about 42%. This is primarily due to testosterone. So what makes male skeletal muscles grow larger than females? It's the amount of testosterone. Muscles actually age. By about 30, we start losing muscle mass. So it's almost like we have a peak up to about 30, and then we're going to have a slow decline in muscle mass. We call this sarcopenia. This is not the same as disuse atrophy that we talked about earlier. This is just in the normal course of life. As your hormone levels decline, you're going to have loss of muscle mass. But we do know that regular exercise reverses some of this muscle loss. Last slide has to do with muscular dystrophy, specifically something called Duchenne's, where you're missing this dystrophin protein. All of the other parts of muscle mechanics are working. So you still have the thick and the thin, and you have the myofibrils, and you have the calcium and the troponin. You have all the necessary parts. You're missing, this is the connection. This connects the action inside the sarcomere or the myofibril to the entire muscle, the whole muscle. There's no cure for this, and usually the patients die of respiratory failure because you need several muscles in order to breathe in and exhale. Well, that's it for Chapter 9. Please focus on those steps involved in the muscle action phases.